Bismillah Rahman Rahim. Welcome back, friends. Welcome to another video lecture by Kame Biology. In this video lecture, we are going to talk about nucleoid or prokaryotic genetic material. Okay. Now, this is another video lecture which is also regarded with the cell. Okay. Now, it is actually presented by me. Means my name is Muhammad Kamil Khan. Okay, and this is my channel name, Kame Biology. So let's start. First of all, we will start from the introduction. Okay, so the first point says that prokaryotic and eukaryotic are different from each other due to their genetic material. Whenever you want to distinguish between prokaryotic and eukaryotic, that's about their genetic material. Okay, now the next point says that eukaryotic cell have 2 or more chromosomes in nucleus, as eukaryotic cell have 2 or more chromosomes in nucleus with the membrane bounded organelles. Now we know that eukaryotic cells are eukaryotic cells contain a nucleus with the membrane bounded organelles as well. But on the other hand, if we see the prokaryotic cell, they don't have nucleus, well developed nucleus, and they don't have the organelles. Okay, so when they don't have the organelles, they don't have the nucleus, so definitely they will contain the genetic material which we call that nucleoid. Now the next point says that when prokaryotic chromosome are located in irregular shaped region, then we call that nucleoid. Okay, this nucleoid, or we also call it nuclear body, chromatin body, or nuclear region. When the prokaryotic chromosomes, the prokaryotic chromosomes are usually found where it is usually found at the center, but it can occupy the whole cell portion, and it's very complicated and complex. Means this nucleoid are very complicated and complex. Now here I will show you the nucleoid diagram or nucleoid structure, that how the nucleoid will be present. Now as you are watching, inside the prokaryotic cell, the nucleoid will be in a circular form. Means this is the double stranded DNA, and it usually in a circular form in prokaryotic case. Okay, now second we have this is also means nucleoid. So one thing more that I must tell you, the genetic material of the nucleoid, it contains chromosomes which is irregular in shape, double stranded but irregular shape, and nucleoid may also be the plasmid as well. Now second point, now another point says that usually prokaryotics contain a single circular of double stranded DNA. As I show the diagrams to you, as I showed the diagrams to you, that prokaryotics contain what? Prokaryotics contain a circular double stranded DNA, but some have a linear DNA chromosome. Not only all prokaryotic cells have the circular DNA, but some of them are the linear chromosomes. So, for example, in case of Vibrio cholera and in case of Brolia burgdorferi. Now this Vibrio cholera are the causative agent of the cholera, and this Borrelia burgdorferi are the causative agent of Lyme diseases. Now such microbes have what? Such microbes have more than one chromosomes, but usually prokaryotic have only one chromosomes material. Means they have a chromosomes material but irregular in shape. Now next point says that both electron and light microscope are used for the understanding nucleoid structure and function, especially during active cell growth and division, because it is not possible for us to examine the nucleoid structure when the cell are not ready to growth or division. Okay, you should have to keep one thing in your mind: whenever the cell are dividing or growth, then you will easily check the nucleoid structure both in electron and in light microscope. Now this is the diagram of the light means electron microscope. It is connected with the computer. We can we can easily see the diagrams, or we can easily see the pictures on the computers. This is the light microscope. Whenever we want to study the nucleoid structure with the light microscope, we usually stain we will send the slide, and then we will easily see the structure of the nucleoid on the light microscope. Now next point says that nucleoid has a fibrous appearance in electron microscope. The fibers are probably DNA. What does it mean? If we see the nucleoid structure under the microscope, so it is usually in a fibrous shape. In the next slide I will show the diagram. Okay, so it is usually in a fibrous like shape, and such fibers are what? Such fiber are DNA. Now next point says that during cell division the nucleoid has a projection contain DNA that transcribed to produce messenger RNA that extend into the cytoplasm plasmic these projections. What does this point means? This point says that during cell division what actually happen, the nucleoid will be appears. Once the nucleoid is appears, then we can see a projection like structure, and such projection like structure will contain DNA. Now such DNA will be transcribed into messenger RNA. Okay, once they transcribe into messenger RNA, then they will be expelled out, and or we can say they will be extended to the cytoplasmic area. Now this projection, I am talking about the messenger RNA. The messenger RNA is actually the projection which are usually transcribed by this DNA, and it is released into the cytoplasmic area. Such messenger RNA then used for the protein synthesis because they contain a codon which are specially responsible for the protein. Now next point says that more than one nucleoid can be observed within a single cell where the genetic material has been duplicated, but cell division has not yet occurred. Not only the prokaryotic cell have only one nucleoid, we can observe more nucleoid as well, but it doesn't mean that when the cell have more nucleoid, so it will be divided. Now without division the cell will have more nucleoid. Okay, so the nucleoid will be duplicated, but it doesn't mean that the cell will divide. Now this is the diagram of electron microscope. When you see the nucleoid, now this red one represent what? These red one represent the nucleoid structure. Okay, this is the cell. The cell is divided, then you can easily check what? You can easily check the this means nucleoid. Nucleoid can be isolated. When we treat the cell with the different kinds of chemical, then we can easily isolate the nucleoid. Nucleoid are composed of about 60% DNA. This is very important. It is composed of 60% DNA, 30% RNA, and 10% of the rest of the protein. So these are the important material due to which the nucleoid are composed. Next points we have: in E. coli the closed DNA circle measures approximately 1400 meter or about 23700 times longer than the cell. This is very important. The E. coli, the E. coli chromatids, or we can say the E. coli nucleoid, are very long and very complex, and it is approximately 1400 meter. And what does it mean? It means that it is 23700 times longer than a cell. I will show the diagram in the next slide, then you will check it. Now next point says, unlike the eukaryotics and some archaea, bacteria don't have don't use histone proteins to package their DNA. If we see the eukaryotic or archaea, so in eukaryotic cell and archaea, they have what? They have histone protein. Due to this histone protein, the chromosomes will be packed and the chromosomes will be appear into a DNA like shape. But such kinds of such kind of structure will be not observed in the bacteria. Now this is the E. coli structure. These are the fibers. Okay, these are what? These are the fibers of DNA. These all are the DNA fibers, and all these are nucleoid as well, and all these are what? Chromatin as well. So that's all about the nucleoid. Thank you so much for your watching. I hope you understand about that video. If you like this video, so please make sure to hit the like button, share this video, subscribe the channel to get more interesting video lecture like that. Take care. Bye.